Hi, I'm Kinalis, and I'll be discussing the use of programming and the applications of it through microbiome analysis. So starting with programming, today we have many devices and technology that all relate back to the art of programming. And when you write that first line of code, you often think, why do I write this, and what's the purpose of programming? And the bottom line is to solve a problem, to make the problem that you're solving easier. And what we find as we program more and more is that not only are we solving the problem with programming, we're solving it more efficiently and quicker. And as we use programming more and more and we develop it more into our lives, we find more and more uses of it. So in our lives today, some ideas of programming can start with the calculator, where you have the calculator that takes all these calculations that would otherwise take a very long time for a person to do. And then you develop that idea of more and more programming, and then we move on to things such as a GPS, which not only uses the idea of a calculator, but more sophisticated programming, such as using satellite images. And then as you advance even more, you have other things such as the smartphone, which not only includes the calculator, but the GPS itself, and it has its application. So those are everyday uses of programming, but how about going even further with it, and specifically in the research field? Well, in recent years, the idea of programming has been very apparent, specifically by biologists, in terms of how they can use programming to analyze their data and what they can use it for. And in recent studies, they found that programming was used as a way to use a software called SPICE to replicate the bodily functions of an organism. And what this did for the researchers was allow them to monitor the software as if it was an actual organism, to take data down on it rather than doing it on the actual organism itself. And similarly, another research group use an algorithm as a way to replicate the idea and functions of living tissue, to monitor the living tissue in an organism had they actually done it on that organism. And what both these researchers have in common is that they're using programming as a way of data collection, as a way to get more data and sort through it in the amount of complex data that they already have. Going off this idea of taking programming for complex information and narrowing it down and making it easier to process, we come to microbiome sequencing, which is essentially taking the community of bacteria on a given sample, analyzing the DNA sequences of those bacteria, identifying them, and see what their effects are on the ecosystem and other organisms around them. And with this process, there's no definitive way of actually performing the microbiome analysis that's the most efficient or a quick one. There's no actual one that's preferred overall. And the reason for this is because of the fact that the larger number of DNA sequences in a sample can go from a couple hundred to a couple thousand to even more than that. And what you find with all those DNA sequences is not only are there such a large amount of DNA sequences, the calculations from programs and having to actually narrow them down and then identify the bacteria based off those sequences takes an extremely long amount of time. And what this research and what I want to try to attempt is to not only attempt microbiome analysis with a new method, but to see if it's actually possible. And because of the fact that this is a new method, and that I'm trying to solve the issue of the fact that there's no definitive method already, it serves as a problem-oriented study in which the process that I complete, the product isn't so much the solution, but the actual process is the solution itself. So just to start with the idea of how this would work, there's three basic parts that I'll show an image of in a second. And you start with the DNA sequences and raw text. And what happens is you take those sequences, you manipulate them by using programming and commands on them to give you an illustrated product. And how this starts is you start with the QIIME website, which is the website that holds all the DNA sequences. And from there you move all the sequences onto a common platform. In this case it's called an online notebook called Jupyter. And in that notebook it runs parallel to Python programming, which will allow me to initiate different types of commands to manipulate that data how I so please. And in the end, I want to attempt to take all those sequences, identify them, sort them, and then produce a heat map of the bacteria that I will show you later on. So again, this is the image of the process. You start with the raw text. The Jupyter Notebook is this pathway between getting from the text to the actual illustration of those bacteria from those sequences. And to start with Jupyter itself, this is what the actual layout looks like. You can see in there's little tiny text boxes called cells. In each cell you can run a command, and based on what command you want to do and how you want to specify for specific sequences, it will run that command for you. So to start with the process, the sequences are first on that website. You need to take those DNA sequences and put them on the Jupyter Notebook so they can be manipulated. Using a simple command that looks sophisticated, all the computer is really saying is go to this website, take those sequences, put it on the Jupyter. That's the first step. Once those sequences are in the Jupyter, they're ready to be manipulated and tested for, but the thing that you first have to consider with these sequences is, as again, there are many sequences, And with each sequence, the amount of text that they take up and space that they take up is very large, and the first 12 lines of each DNA sequence is really the only amount that you truly need when taking this into consideration, because of the fact that the first 12 lines are unique enough for each DNA sequence to be compared to different bacteria. And had I not had cut the first 12 lines, then this process would have taken possibly even longer, or would not have worked with the service that I was working on. Once the sequences are cut and ready to be manipulated, they're all taken from their own additional files that were put onto the Jupyter and put into one large file, so they can all be manipulated and compared to each other later on. Once they're in that large folder in the Jupyter Notebook for comparison, what is then done is each one is assigned an ID, a certain number for each sequence. The sequences are then compared to each other, and if they have a 97% similarity or matchup, they're considered as one sequence or one bacteria. Because after they are ID'd and then matched together, then they're then compared to an online database, which determines which bacteria it is based on taxonomical units, which can start as broad as the kingdom, as specific as the genus category. Once they are identified, ID'd, and then sorted properly, you have all the bacteria named for those sequences. The only step left is to actually take those bacteria and produce them on a heat map for people to actually analyze. Now, this was actually produced by me. I took those sequences and I was able to generate the heat map. This is at the kingdom level, and as you can see, you have 3 different boxes, and each box is a different area of each samples, and those boxes are collected in the same area. And looking at these different colors, those represent the relative abundance of each bacteria in each given sample, and you can see the table of the different colors that represent those bacteria. And what you can see is, based on the location that they're in, their relative abundances are relatively the same. You can see which bacteria is in large amounts compared to the smaller amounts, and in each series, they're all similar, except for the exception of this one. Now, I mentioned before that when classifying the bacteria, not only does it go from the kingdom, it can go much more specific. And as you can see, going to the genus level, you can see that the distribution of bacteria abundance, when narrowed down more specifically, is much more diverse, as seen here, which shows much more bacteria based on how they're classified. And to give you an idea of how much bacteria is actually present, it's such a large amount that you could barely fit on the page. So, what does this data prove? Well, for one, the fact that I was actually able to get the heat map shows that the process that I ran to do the microbiome analysis was successful. Looking back on the data that I was showing you, being able to identify that in those 3 different groups that the relative abundance was about the same allows you to understand that I was able to interpret the actual data and that it wasn't just incorrect or incoherent. Lastly, the fact that it can go from kingdom as specific to the genus category shows that this process is not only successful, but that is very useful in terms of how in-depth that researchers want to go in terms of the bacteria that they're studying. In terms of implications for the future with the use of Jupyter, I expect that if the Jupyter method that I used is perfected in terms of how it's run, it can in the future be used as the most quick and viable way for people to understand microbiome analysis. And not only this, but if in the future it's perfected, they're planning, the Cold Spring Harbor laboratories that actually ran this method, they're planning to make it autonomous, meaning that all the manual commands that I ran would be done by the computer itself, and in the end product, whatever users using that microbiome analysis method would just have to input their sequences, and the whole process would be done for them. And what this leads us to believe is that this method, if used in the future, will become one of the quickest and most successful. And just to give you an idea, from Cold Spring Harbor Laboratory, there are 4 methods of DNA analysis and barcoding that are currently available. And in the future, if this process was able to be run and perfected autonomously, then it could become that new line for a part of the other 4 DNA methods.
Okay, thank you, Nick.
Okay, how is the method you chose aligned with the purpose of your research, and which methods did you consider and reject?
So in terms of how I went about doing this research, again, the overall topic was there was microbiome analysis. The issue was that there was no definitive or proper method that was used overall. The method I chose to do was part of a solution, or solving the problem of that study. So it was a problem-oriented study or methodology that I took in terms of looking at this project and seeing what I could do to actually perform the microbiome analysis. In terms of other ways of looking at it and performing different types of studies, I thought the best way of actually looking through and seeing what method would actually work was the best way in terms of studying it, only because of the fact that by performing this solution, showing that the method works, solved at the issue that I was planning to do.
What may be some real-world implications or consequences, like the influence on others' behaviors, decision-making processes, or discoveries that are related to your findings?
So as I was explaining, I showed you that the Jupyter method that I used was actually successful. And being that it was, if it's implicated into the research field specifically by other biologists that are studying microbiomes, specifically such as Cold Spring Harbor, what it allows them to do is to study the organisms or the samples that they're studying currently at a much faster rate. An example would be the DNA sequences that I used were actually local Smithtown Bay mussels, which in terms by doing this method, I was proving or helping to disprove the idea of whether or not the DEC was saying if the mussels on certain locations were safe or not. And by doing this method, it shows that you're able to determine whether or not that's true, because based on the location that you saw on that heat map, their microbiomes were in fact different, which could propose that the mussels really are different based on their microbiomes.
Thank you.
In which specific part of your research process was your consultant most helpful, and how was he or she most helpful? What did you learn from the consultant about your field of research?
My consultant came from Cold Spring Harbor Laboratories and actually worked on hand with the Jupyter process, worked in the Jupyter notebook and the Python programming, and was familiar with the Python programming. Going into this research, I lacked depth of understanding how to use the commands that came with Python programming, so with his help, he taught me which Python programs to run, what they meant, why I had to start a certain program before another one, and which values to use in those programs in terms of how to run the servers properly. And in terms of what I learned from him is that Python programming itself is a very unique and difficult to understand at first programming language, but with patience and understanding that the capabilities of it can really be a pioneering technique for microbiome analysis.
Thank you.
Thank you, Nick.